Okay. All right, we should be live. All right, so thanks everyone today for joining our micro seminar to hear Nicole tell us about the fantastic metabolisms of archaea. And for anyone inside the Hangout, I'll remind you that we'll keep you guys muted and webcams off. A seminar to hear Nicole tell us about the fantastic metabolisms of archaea. Okay, now I'm not playing on YouTube anymore. And so Nicole had done her PhD at Wisconsin-Madison and then postdoctoral work at the University of Illinois, Champaign-Urbana, and then joined the faculty at University of Nebraska-Lincoln, where they have the most fantastic, oh my goodness, what's it called? The speakeasy downtown, which is fantastic. If you guys are in Lincoln, Nebraska, you got to go check it out. So she can take it away, Nicole. If you want to tell everyone about your talk and share your screen, that'd be awesome.
Okay, so does that work?
Yep, that looks great.
Okay, great. All right, so thanks again, Jen, for the invitation, and thanks everybody for tuning in live and through the recorded YouTube channel. I'm very excited to have the opportunity to tell you about some of the really cool work that we're doing in my lab, where we're looking at manipulating metabolic efficiency. And what's really another big take-home message is that we can learn a lot from studying kind of weird microbes like the methanogens that we study. What we learn from them really tells us about kind of general big rules in biology that we can apply to all organisms. So this is the TLDR slide. Basically, it's all about the electrons. That's the main take-home message. Redox reactions are essential for life of all types. And if you can understand how electrons and carbon especially flow through a cell, any cell, you can really control ultimately all the chemistry that the cells will do. And it's really important to study extremophiles like methanogens to learn the endpoints of biology, as it were. What are the limits to biology, and be able to fill in all of those points. And ultimately, you can use that knowledge to eventually solve real-world problems. So first, I want to take a couple minutes to tell you that I'm a microbial physiologist. And a lot of people have kind of confusion about what that really means to be into microbial physiology. The main questions that we want to answer are, what do cells need to grow? How do they grow? Like, what's the mechanism, the biochemistry, the gene expression, and all those sorts of things? How do they work together? And can we use that knowledge to make them grow better? And what tends to be confusing about it is microbial physiologists take a really interdisciplinary approach. We'll use usually whatever tools we can to understand how our favorite microbes grow. And it's really a very fascinating, interesting place where biology, chemistry, physics, and all of these things intersect with the study of microbes. So we tend to try to pull from as much scientific information, math, and anything you can to gain insight. And microbial physiologists often will spend a lot of time trying to define what's necessary and sufficient for their system or their pathway of interest. And we tend to appreciate sort of the minimal point. So the kind of the rule of parsimony, what's the most direct way to get from A to B to get this phenotype to occur, or to get this chemical transformation to occur for this step in the pathway. However, there is a lot of redundancy in biology. And it's important to realize that the goal of biology is not just to be the most efficient to go from A to B, but just to exist. So there's really a lot to appreciate in biological diversity, that there are many ways from A to B. And that's where the beauty of biology is evident. And it's important as a microbial physiologist, or one of my favorite thing to think about, is that some of those ways are more efficient than others. So the organisms that I really am most interested in studying are the methanogenic archaea, what we call methanogens. If you are a microbial physiology nerd, you probably know about methanogens. They derive all their energy to grow by reducing C1 compounds like carbon dioxide or formate or C2 compounds like acetate into methane gas. And they're strict anaerobes, and they're key players in the global carbon cycle. About 4% of carbon on Earth goes through methane, is estimated to go through methane every year. That doesn't sound like a lot, 4%, but it amounts to about 2 gigatons worldwide. So if you looked at a lake in Canada when it was still frozen over, when you can see all these trapped gases that are produced by the submerged anaerobic microbes and consortia underneath that lake that are continuing to consume and mineralize all of those CO2 and methane, it actually is quite a lot of net influx of CO2 and methane that can be produced by methanogens in these sorts of environments. But they don't just produce greenhouse gases like methane and CO2. Methane actually is a rocket fuel. So that was part of the trailer that I mentioned earlier. Methane, either through fossil sources or the renewable type of methane that's produced by methanogens, can be compressed and used to power rockets. They can also be used as a transportation fuel or for electricity, and burning methane is much cleaner than diesel. Not only do they make this rocket fuel, but they also make life-giving medicines. It turns out that about 50% of people on chemotherapy will be administered this drug, Mesna, that helps them tolerate their regimen. And Mesna is actually the coenzyme M cofactor that was discovered in methanogens. So they're incredibly important, fascinating organisms. They dominate in low sulfate sediments, such as in this idyllic kind of mountain stream. But methanogens are also often the dominant archaea in the human gut, the termite gut, and then the rumen of cattle, so these natural anaerobic environments. So there's so many great reasons to study them. They're part of our natural flora. They're part of the global carbon cycle. They make this renewable natural gas rocket fuel. And it turns out every year there are more and more methanogens being discovered. It turns out that we're starting to find maybe butyrate oxidizing methanogens, which is really exciting. And it's maybe a little bit controversial right now, but there's a sense from the phylogenetic world, people trying to identify the last unified common ancestors of all life on Earth, that perhaps the first eukaryote was also a methanogen. So we could actually all really be methanogens. And it'll be exciting to see how that develops. The organism I'm primarily going to talk to you about today is Methanosarcina acetivorans. So I mentioned that methanogens usually dominate in low sulfate environments. Well, this organism was isolated off of Scripps Canyon in La Jolla. So I don't have a picture of Scripps Canyon. This is my picture of Hanauma Bay because I'm originally from Hawaii. And so in an environment like this, where you actually have a lot of sulfate and sulfate reducing bacteria that you think would be more competitive, more energetically efficient, and more abundant in these environments, nevertheless, you find methanogens like Methanosarcina acetivorans. This methanogen is able to synthesize cytochromes in contrast to most other methanogens. And they are also, and we think the ability to synthesize cytochromes is a part or potentially a cause for its ability to use multiple carbon sources to grow. So most methanogens that we've been able to grow in culture can only use one or a few simple carbon sources, and these guys can use multiple ones. So they're more metabolically flexible than other methanogens, and that makes it easier to study genetics and physiology on them, in my opinion. So despite the fact that they're these exotic anaerobic organisms, we can actually do just about everything you can do with E. coli with methanogens. It's just that you have to do it in an anaerobic chamber under very strict anaerobic conditions. We have to treat our media specially. We have to use special glass and butyl rubber stoppers, but we can pretty efficiently move genes around and delete them as we like through various different tools. So again, if you are a major microbial physiology nerd like me, Methanogens are just the bee's knees. They're the best. And one of the reasons is because you could consider them to be sort of energy extremophiles. So one of the main concepts that was proposed and described elegantly by Rolf Tauer many years ago was that for microbes to exist, they need to extract energy from the chemicals that they're growing on, obviously, and that the amount of energy that they're able to extract can be somewhat predicted by the Gibbs free energy equation. And if you look at methanogens and the substrates they can grow on, methanogens that can grow on acetate, for instance, on the left, are using a growth mode that's really, to that far extreme of being really entropically driven or retarded by the enthalpy of the reaction that they're carrying out. So that's, way to the left on this scale. And in the contrast, when methanogens are growing on hydrogen CO2 as electron donor and acceptor, that process is entirely enthalpically driven. So that delta S term is really the driving factor in the amount of energy that they're able to extract from that growth mode, you could say. And when methanogens are growing on something like methanol or trimethylamine, that really falls right in the middle near, between aerobic respiration and fermentation. So by studying methanogens, and especially the Methanosarcina methanogens, which can grow on all of these substrates, you can really, by changing the growth mode, understand how the chemistry affects the efficiency of how much energy they're able to extract from their substrates. So that's really pretty fascinating to me. So I wanted to ask the question, can we control the flow of mass and energy, so carbon and electrons, through these methanogen cells by manipulating the electron transport system? So here I'm showing you the Methylotrophic Methanogenesis pathway. So this is how acetivorans grows on methanol as a sole carbon and energy source. And it's important to realize that the electron transport system, like what I'm showing you here, but also in any other organism, is 99% of the chemistry that's occurring in the cell. It takes a lot of electrons, it takes a lot of protons or ions moved across the transmembrane or moved across the membrane to produce enough ATP to make an amino acid, for instance. So a lot of people who will study very important phenotypes or physiological properties like chemotaxis or cell signaling or biosynthesis of peptidoglycan and all of those things, very important for cells to grow, but really that's this tiny little biosynthesis arrow way at the left. 99% of the chemistry is going to go through the electron transport system. So for methanogens to grow, they will take this methanol compound and attach coenzyme M, which is a small thiol. This is the same thing as that mesna drug I talked about that people receive. And they produce methyl coenzyme M. This methyl coenzyme M is oxidized to carbon dioxide, and these electrons are extracted and carried by these electron carriers, ferredoxin and the coenzyme F420, which is a really cool diazoflavin cofactor. The electrons from here go through the electron transport system. The green steps are where we have proton or ion translocation across the membrane in such a way that it favors future ATP production. And those electrons flow down the electron transport chain, ultimately reaching the coenzyme M, coenzyme B heterodisulfide that's produced in the last step of methanogenesis by methyl coenzyme M reductase enzyme. So the electrons are extracted from CO2, flow around, and are used to reduce additional molecules of methanol to methane gas. So yes, this is why people go a little bit nuts about methanogens. It's because they make their own terminal electron donor and their own terminal electron acceptor. So instead of using, respiring a metal or respiring oxygen like people do, they're actually respiring this heterodisulfide here, which is the terminal electron donor for this last step to make methane. That's the terminal electron donor, and this disulfide is the terminal electron acceptor for the electrons that are flowing through the membrane there. So that's pretty cool and unique and exciting about methanogens. So if we want to be able to make methanogens grow better or faster by tweaking the electron transport system, we're really going to target all of these steps in some way or another, and that's really what my lab has been doing in the past few years. So the first target is going to be the heterodisulfide reductase enzyme. And that's because this is the terminal acetivoreductase in the cells. This enzyme activity, reduction of this heterodisulfide to the free thiol forms of coenzyme M and coenzyme B, is essential in all methanogens. They don't have enough energy to keep making more of their terminal electron donors and acceptors, so they have to recycle it. And this is the enzyme that does that. So, and it's important to realize that there are these two classes of heterodisulfide reductase in Methanosarcina acetivorans, this HdrABC and this HdrED version. So the hdrABC is a non-energy conserving heterodisulfide reductase because the electrons are just going to flow from the electron donor, which could be usually thought of as being a ferredoxin or F420, to the heterodisulfide. And it turns out that they will bifurcate and possibly, also not bifurcate electrons to co-reduce low potential ferredoxin. So it's a really pretty fancy, awesome enzyme complex, but it doesn't contribute to translocating ions across the cell membrane. So it's not energy conserving. It doesn't contribute to ultimately ATP synthesis. In contrast, you have the cytochrome containing hdrED, which takes electrons from reduced methanophenazine in the cell membrane, which I'll talk about a little bit more later. But by oxidizing methanophenazine, it actually is participating in translocating protons to the outside of the cell. So this is an energy conserving heterodisulfide reductase enzyme. And it was really interesting when we looked at the genomes that these methanogens have not only two types of heterodisulfide reductase enzyme, but multiple alleles for these genes. And so when we were initially trying to understand this, you know, there's several hypotheses. Well, you know, why would this non-energy conserving one be important, or potentially non-conserving energy conserving hdrABC version be in the Methanosarcina when they have an energy conserving one? That doesn't seem to make sense. So either it could be non-functional or it has another function, you know, what's going on here? So I did some transcriptional fusions, and indeed all of these loci were transcribed into RNA, and the HdrED, the energy conserving one, was expressed under all of the growth conditions that we tested. The different other loci were expressed to different levels, and there was some substrate specificity there. And this one particular hdrABC locus, where all of those proteins are in one operon, seemed to be expressed under methylotrophic substrates, but not under not while cells were growing on acetate. So it seems to be the methylotrophic heterodisulfide reductase enzyme. But you know, so they're all expressed. Are any of them essential like we would predict, or are all of them needed, or only some of them? So I put a tetracycline controllable promoter in front of each of these loci to shut off gene expression unless we added tetracycline to the media to cause de-repression of gene expression. And so I made all of these different strains separately and looked at how they grew. And sure enough, if HdrED was repressed, the cells would never grow. And so I would eventually see growth in these tubes, but when you went back and sequenced the promoter region of these loci, they had all mutated to a highly expressed version of the promoter. So that's really good evidence that they really need this HdrED to make ATP, as we would expect. The other ones were not essential. But was pretty confusing about this is that while there was a long lag, they eventually would come up and they would grow at a slower rate than the parent strain. But ultimately, they would reach the same, growth density, the population density. And indeed, the hdrABC that looked methylotrophic indeed didn't appear to have an effect on acetate. So they seem to be growing slower, and sure enough, when we look at the rate of respiration, so the rate of methane that was produced on methanol, it was half the amount that was produced by the parent strain. And on acetate, there was really no difference, and that's consistent with what we saw, that it doesn't seem like it's important for acetate, growth on acetate. So the conclusions from this kind of earlier work was that, okay, HdrED, the energy conserving one, really is essential, and that it was really pretty surprising that this organism uses both flavors of heterodisulfide reductase simultaneously. And we saw that the one was, hdrABC was a methylotrophic heterodisulfide reductase, so there's some substrate specificity. But nevertheless, without the hdrABC version, it seems that the reduction of the heterodisulfide coenzyme B, coenzyme M, was rate limiting through hdrED, saying that it's not good, and it's to the cell, it's not good enough, it's not fast enough on its own. And that's also borne out by when we look at the phylogenetic distribution and homologs of the HDR genes in methanogens and their sort of more closely related bacterial cousins, you could say. So in all of these methanogens, not only do they have multiple versions of the hdrABC genes, so each organism will often have multiple copies on their chromosomes, and they're also really widespread throughout the methanogen world, and then also in the geobacter, soil microbes, sulfate reducing bacteria, and the chloroflexus, for instance. So this enzyme and its sort of cousins have been really important in the energetics of lots of different organisms. And there seems to be a pressure for, like, an evolutionary pressure to expand the utilization of the hdrABC genes. So why would that be if they're non-energy conserving? Why would you pick to use hdrABC over hdrED and acetivorans? And the only thing I can really think of is to go faster. Because if you're using hdrABC to reduce the heterodisulfide, it's essentially, one step from your electron donor to the disulfide, whereas with hdrED, you have these other mediators in the way, these membrane protein pumps and methanophenazine. So it's a more parsimonious route, even if you're not contributing to an ion motive force. So we kind of call that a futile cycle, because it seems to be bypassing what would be useful for the cell. But if it's going faster, that might be useful for us. So could we maximize the rate of methane production by tuning this futile cycle? So to do that, we put another copy of the heterodisulfide reductase hdrABC on the chromosome at an additional site. So we created this plasmid, integrated onto the chromosome, and we're comparing the physiology, so the growth kinetics, so on and so forth, between the parent strain, our knockout strain that we have, a strain that produces or has two loci, and then one where we're complementing back the mutant. And indeed, what we saw is that we could complement the growth rate phenotype. So the cells are able to grow just like the parent strain when we had this additional copy of the heterodisulfide reductase locus on the chromosome. And very satisfyingly, when they're growing on methanol, the ones that are overexpressing hdrABC appear to make methane about 30% faster in cell suspensions than the parent strain, and a lot faster, obviously, than the deletion mutant strain, which was half the rate of the parent. And just as we predicted from all of the other growth and transcription data, there seems to be no benefit or effect on acetate, suggesting that, you know, it's still substrate specific the same way it was, you know, previously in the transcription assays. And then we also did enzyme assays to really check the activity, how much heterodisulfide reductase can be reduced per unit time from these cells, and we saw that our overexpressors were indeed overexpressors. So what we would predict is that, well, if we have this futile cycle, that the cells should be, you know, they're faster, but they should be less efficient. So if we compare the rate of methane production, so this is the respiration product, methane, versus the HdrABC enzyme activity, we see a very nice linear correlation here on the left. And it matches very well with our deletion mutant, where we have this heterodisulfide reductase enzyme completely knocked out. And if we look at the growth rate, so how fast the cells are doubling compared to the methane production rate, we see that it's not a nice linear relationship, which is not that expected, not unexpected. Basically, because the heterodisulfide reductase rate is limited in the HdrABC mutant, the growth rate is also slower, but you get to a point where more HdrABC isn't necessarily better, but it's also kind of surprising that it's not a whole lot worse either, suggesting that there's basically, you know, some level of prioritization for biomass production, but then at a certain point, you have to work a lot harder to get that to diminish. And we haven't reached that even with our overexpression strain. So if you look at the biosynthetic efficiency, so the amount of biomass that's produced per mole substrate consumed, we see actually, again, a really nice pattern that we have, you know, a certain efficiency for our parent strain, and it's much higher for our deletion strain because we've gotten rid of this futile cycle. And then the more expression of hdrABC we have, the less efficient the cells are. And then again, so that's plotted out here in a scatter plot, and it's a very nice linear correlation. So there was a lot of, you know, assaying enzymes in electron transport chain and counting number of protons pumped is incredibly difficult. There is data out there, and it's really, I think, one of the hardest kind of biochemistry that anybody could do, because you have to have intact cells, but yet you want to try to have things purified to understand. And so we had, it wasn't as simple, you know, it's not so simple to just assay everything and ask, well, where exactly are these electrons going? So you have to actually resort to modeling at some level. Once you know certain ratios that people have measured, you know, at a certain level, you have to start modeling and seeing how it matches with the observed biology. So that's what we had to do here. There is some controversy about how well pumps work and exactly the route that the electrons were kind of flowing in these cells. So we actually had to draw out 22 different models of all the possible ways that the electrons could be flowing in these cells when they're growing on methanol. And sure enough, there was only one model that really matched the observed biology. So if we use these models to create a white ATP yield and calculate out how many protons were, you know, mathematically how many protons were translocated per mole methanol, and compare that to the actual observed phenotypes here on the right, there's only one of these models that matched, and this was that our hdrABC is indeed not really contributing to energy conservation, and it's making these cells grow faster and produce methane faster, but it's not really contributing to energy conservation. So what we think is that using a futile cycle really works with case strategists. And these are organisms that maybe sacrifice efficiency for speed. You can think about it like the tortoise versus the hare kind of a strategy. Our strategists like acetoclastic and hydrogenotrophic methanogens that are really on these energy extremes of life, they're going to try to be, it seems as if they do try to be maximally efficient, but the sacrifice for that is they're only going to go so far. They're going to grow at the fast rate that they can grow at and be very efficient, but they are less malleable and less flexible than the K strategists, which maybe have more genes, more ways to route their electrons, perhaps, like the Methylotrophic Methanogens. And so they can, we can actually use the enzymes to reroute these electrons to get them to be more or less efficient. And so that's actually pretty exciting to me. So, you know, using hdrABC, we made energy conservation less efficient, but that ended up making methane production rate faster, which is great if you want to make renewable methane. But can we also improve the efficiency of energy conservation? So the flux of electrons and through HdrED, essentially. So that's the essential heterodisulfide reductase that helps to produce a transmembrane ion gradient towards ATP. Can we get them to grow faster and better by manipulating that step? So there are two ways to make enzyme reactions go faster, that are two main ways. One is to increase the concentration of enzymes. The problem with that is that hdrED is already pretty highly expressed in Methanosarcina acetivorans. The other way is to increase the concentration of substrates. In this case, that would be reduced methanophenazine. Methanophenazine is this molecule shown in A. It's this tricyclic phenazine ring that has a polyprenyl tail on the end of it. And it's functionally similar to a quinone, and quinones are membrane electron carriers in about half of all bacteria and most eukaryotes. And so it's functionally similar to this quinone in that it's shuttling electrons from an oxidoreductase to its terminal oxidase, and it does the same thing there. And so in Methanogens, depending on the growth mode or type of Methanogen, methanophenazine is reduced by these various oxidoreductases such as FPO, RNF, or VHO. And when they're reduced, when methanophenazine is reduced, it actually takes up a proton from the inside of the cell. And when the reduced methanophenazine donates its electrons to hdrED, those protons are actually released to the outside of the cell. So there's a net translocation of protons across the cell membrane. And that's how hdrED contributes to energy conservation in these organisms. So could we manipulate the concentration of methanophenazine? Well, we realized we don't really know how much methanophenazine they make. So we decided to measure that. So we developed a kind of a nice method that tweaked the method developed by Abkin, as well as thinking about the quinone extraction protocols that people had. And we extracted the methanophenazine from Methanosarcina acetivorans growing on methanol throughout the growth phase. And we were really kind of fascinated to see that the amount of methanophenazine seemed to change. So in earlier exponential phase, there seemed to be fewer molecules per cell of methanophenazine than when they hit stationary phase. And then, you know, the rate or the quantity of methanophenazine changed while cells were in stationary phase, but at a much slower rate. And that was pretty exciting because, again, people had this concept that methanogens, you know, energy extremes, yada, yada, they're so efficient. And if methanophenazine is so important in Methanosarcina acetivorans, why would it change the concentration? You think that it would pick the optimal concentration and keep it at that. And so that was a pretty surprising result. And we wanted to ask, well, how does this work in Methanosarcina barkeri then? Methanosarcina barkeri is closely related to Methanosarcina acetivorans, but it uses a hydrogen cycling mechanism, which I don't have too much time to talk about, which is a shame. But anyway, it actually uses hydrogenases to produce hydrogen, and then it sucks the hydrogen back up. And methanophenazine is involved in those oxidoreductase or hydrogenase reactions, and ultimately still used to donate electrons to hdrED. But you have this diffusible gaseous hydrogen that goes outside the cell, and then the electrons flow back into the cell. So its electron transport chain is actually importantly very different than Methanosarcina acetivorans, even though they both rely on producing methanophenazine. And we were interested to see that, and surprised that in M. barkeri, the methanophenazine that was produced was of 2 flavors, and that's not what we saw in acetivorans. The prenyl tail had different saturations. So it seemed like there was a monosaturated form that when cells were in like earlier exponential phase, it was monosaturated. And then as the population reached stationary phase, it seemed like the monosaturated form was turned into a double saturated form. So there's a negative correlation between those 2 types of methanophenazine in the cells. So that got us to thinking that maybe the efficiency of electron transport actually changes in these methanogens. So that was very, likely the case maybe in acetivorans, because the amount of methanophenazine goes up double between exponential and stationary phase. In barkeri, the amount of methanophenazine is lower than it is in Methanosarcina acetivorans, and then we have this weird, prenylation reduction going on. But that was, that's sort of reminiscent, however, to what we already knew about E. coli, which produces menaquinone and ubiquinone, and the ratios of those change, or the amount of the quinones changes in E. coli, and that's been known for a really long time. So it's kind of, as different as methanogens are from E. coli, there are certain functional similarities between the methanophenazine and the quinones. So what does the consequence, or what could be a result of changing the concentration of the quinone or the electron carrier in these cells? Well, I wanted to ask that question, so we just used measured cell parameters like good physiology. We did that and calculated the sort of average density of the electron carrier in the membrane of E. coli, Methanosarcina acetivorans, and Methanosarcina barkeri. And we saw an interesting pattern here. So in stationary phase for E. coli, or exponential phase in E. coli in blue, and it goes up as they hit stationary phase. The same pattern is seen for acetivorans. As they hit stationary phase, the density, so the number of methanophenazine molecules per square micron increases as well. And in Methanosarcina barkeri overall, there's not a huge change, and the methanophenazine levels are significantly lower than they are in Methanosarcina acetivorans. And so we used these numbers to then ask whether or not these electron carriers were close enough to do isopotential electron tunneling. And what that is when you have an electron donor and an acceptor, the rate at which the electrons go from the donor to the acceptor is dependent upon their distance and the difference in the energy levels of the donor and the acceptor. When we're talking about isopotential, I'm talking about the donor and acceptor are the same energy level, but if you get them close enough, an electron can still hop across without an appreciable loss of energy. So they have to be really close. They have to be within about 15 angstroms from an electron donor to an acceptor. It could be methanophenazine, it could be a quinone, it could be an iron sulfur cluster between 2 isopotential iron sulfur clusters, for instance, could be between 2 flavins, so on and so forth. So it turns out that the methanophenazine and Methanosarcina barkeri is never really at a high enough density to do isopotential electron tunneling. In Methanosarcina acetivorans here in blue and this yellow, both in exponential and in stationary phase, the methanophenazine is concentrated enough on average that if it was evenly distributed, there could still be isopotential electron tunneling from one methanophenazine, reduced methanophenazine, to another. And E. coli seems to go from a state during exponential phase where tunneling is not so probable, but as they hit stationary phase, the amount of quinone goes up and it becomes more possible. So here on the gray. So what does that mean? Well, if isopotential electron tunneling is possible, that means that it's possible that the whole membrane can rapidly conduct electrons. So if these electrons can hop rapidly, they can go from a donor to an acceptor with a minimal loss of energy, and it doesn't necessarily matter how far the acetivoreductase and the hdrED are from each other across the membrane. So diagrams of that are shown on the right here. So, and in B, showing that if the electron donor and acceptor are far away and you're requiring diffusion to occur for them to get close enough to donate an electron, then there's a chance that the energy of the electron is going to decay. So, you're losing energy the farther your electron donor has to diffuse to get to the electron acceptor. So if they're quantized, they can hop across, like shown in C and D. And so you lose a minimal amount of energy, and you could get away with fewer molecules of terminal oxidoreductase and terminal oxidase, for instance. So that means is that it appears like acetivorans membranes are near maximally efficient because they are already within the range of tunneling, but Barkeri membranes probably are not. So how do we test that? Despite the fact that tunneling is possible, you can still make it go faster the more you add, up to a certain point, obviously. So we wanted to test and see, well, maybe we could just add more phenazine and see if the rate of growth could be improved. And if so, the rate or the improvement would be greater for Methanosarcina barkeri because it's less efficient already than it is for Methanosarcina acetivorans. And that's indeed what we saw when we added phenazine to our growth media. Methanosarcina acetivorans growth rates had a small increase, and for Methanosarcina barkeri, we saw a 2 times increase in the specific growth rate. Okay, so that sounds, really? So we wanted to test that. So, well, if the opposite was true, so, that phenazine should be helping electron transfer. Well, what if we did something to hurt electron transfer? Like, is it really getting in there, being reduced and participating in the electron transport chain? So we found this molecule, 5,10-dioxyphenazine, which has to be reduced to become toxic. You start to produce radicals that then go and attack protein and whatever else it's close to. So we tested this by adding it to the media, and the prediction would be that if these phenazines were really getting into the electron transport chain, that this should be highly toxic, and it should be more toxic to Methanosarcina barkeri than it is to Methanosarcina acetivorans, because our acetivorans already has a lot of 2-hydroxy, or has a lot of methanophenazine to move its electrons around, and so there's more alternate routes for the electrons to flow, but barkeri is probably going to be more affected, and that's indeed what we saw. Barkeri was much more sensitive to addition of 5,10-dioxyphenazine than Methanosarcina acetivorans. So from this section, it's pretty cool and surprising to see that Methanosarcina acetivorans is more efficient than barkeri at making ATP when you look at the rate and sort of efficiency of electron transfer through HdrED. And that we can increase the electron carrier pool to increase the growth rate. That's pretty exciting. Some Methanogen membranes appear to be electrically quantized and near maximally efficient in the rate of electron transport from the oxidases to the terminal, or oxidoreductases to the terminal oxidase. And very interesting, basically, to see that we can control the maintenance energy of these methanogens and manipulate it by increasing the forward rate of the heterodisulfide reductase reaction through either hdrABC or ED. So we have cases where we can make it go up and down in kind of a futile or a productive way. So the last thing I wanted to quickly touch on is, can we use these concepts of controlling the efficiencies and controlling the electron flow through the cells to hopefully eventually help to solve real world problems? So I didn't talk too much earlier, but we wanted to see if methanogens could also make alternative bioproducts. So making methane and rocket fuel, I mean, that's already incredibly cool. How you can make it cooler than that, it's hard to conceptualize, honestly. I'm a fan. But, you know, we already can scale up, or we already do scale up methanogens worldwide to treat wastewater, for instance. And there's a huge potential there, and that even the U.S. government through EIA and EPA have calculated it out that it's possible that greater than 50% of the U.S. energy demand could be satisfied through renewable methane used by methanogens. So that's fantastic, but can we direct some of that energy and know-how to make other bioproducts to produce, to replace not just fossil natural gas, but maybe other fractions that come from petroleum? And there would be a lot, there could be a lot of detractors to say, well, these guys are already at the edge of life, except that I just showed you that we can manipulate their metabolic efficiency. But of course, in these bioreactors and things, there's going to be competition for substrates with mixed consortia, all kinds of things happening. And, if you're genetically engineering things, there's always phenotype trait stability because things like to evolve, sure. But we wanted to see if we could use methanogens to make isoprene. Why isoprene? First of all, we use a lot of it. It's a $4.3 billion market for isoprene monomer. We use it to make rubber and adhesives, all sorts of flavorings and all kinds of things. And if you gently hydroxylate it, it's one of the main components of gasoline, so isopentanol. And it turns out that methanogen membranes are made of isoprenoid lipids. So they don't release the monomer form of it that we would use to do all this chemistry, but they turn it into the lipid membranes. That's the defining feature of archaea. So all archaea make a whole lot of isopentanol diphosphate, which is one of the precursors to the isoprene monomer. So if methanogens could make anything, this would be a good thing to test. So that's what we did. And I don't have a lot of time to go into this. Is currently unpublished work, but I'm showing our mass balance equation here. So we created, we think, an isoprenogen, which is very exciting and cool. We can debate about the name. I'd be happy to hear. But by engineering these organisms to make isoprene, we have allowed them to bypass that penultimate CO2 step to produce the carbon dioxide, and instead produce isoprene in the gas phase. And so what I'm showing here is it's comparing the parent cells to the isoprene producing cells. They're consuming methanol in this case, where we showed this mass balance, although they also will do this on acetate and hydrogen CO2 and other substrates. They're consuming the methanol, and they're not producing all the CO2 that you would expect, but the carbon that's missing from CO2 is ending up as isoprene. They're producing the same amount of methane, and they're maybe a little bit happy. They're happy in producing quite a lot of biomass. And so this is also a key point, is remember when I mentioned that the electron transport chain is 99% of the chemistry that happens in the cell. We're not talking about isoprene that's fixed as biomass. We're talking about, this is the isoprene that is going into the gas phase. So it's more like a respiration product. So it's a much higher flux pathway, and it's a higher yield than we would have, hoped for. And so far from what I've seen, I believe this might be the highest flux and yield for isoprene production, according to what's been published in the literature that we've seen. It's also important to note that, making methane and growing by this route is pretty difficult. So it seems like the cells were happy to make isoprene. Their biomass efficiency is at least as good as the parental strain. So they're happy to make it. It's not like we're hurting them by making this product this co-product that we want. They're happy to do it. So what this all means is that it's all about the electrons. It's really important to realize that redundancy and inefficiency is built into biological systems for a reason. Maximally efficient is good sometimes, but not always. And it's really useful for microbial physiologists to understand when you might want to be redundant or inefficient and when you might want to be maximally efficient. And that we can manipulate this metabolic efficiency to control where carbon and electrons go within the cells. With that, I'd like to thank the Buan Lab members. Everybody's contributed to this work in small and large ways. The isoprene project is worked on by Jared Aldridge and Sean Carr. The methanophenazine project was worked on by Annalise Duclouzeau and Nick Dushenko. And the HdrABC overexpression was done by Jenny Catlett and Alicia Ortiz. And everybody has made, great lab to work in. And we're looking forward to learning a lot more and producing some other cool data. And I also like to thank support for this project. The isoprene project was funded by the Nebraska Center for Energy Sciences Research and the Water Environment Reuse Foundation and the Nebraska Public Power District. The other projects in my lab have been funded by National Science Foundation and the NIH, National Institute for General Medical Sciences, through the auspices of the Center for Integrated Biomolecular Communication and the Redox Biology Center. And with that, I'd be happy to take any questions. So I can't hear anybody. Okay. I didn't see that.